Hello, everyone, and welcome to today's live broadcast, Functional Characterization of Gene Regulatory Elements, presented by Dr. Nadav Ahituv, Professor in the Department of Bioengineering and Therapeutic Sciences and the Institute for Human Genetics at the University of California, San Francisco. We are excited to bring you this educational webcast presented by LabRoots. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want, at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window, and type your questions in the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that we'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Please join me in welcoming Dr. Nadav Ahituv. We'll now turn the presentation over to him.
Hi, and so it's a great pleasure to be speaking here today. And my lab is interested in enhancers, which could be sort of simplified as being termed the promoters of the promoter. So, for example, right here we have a gene that's expressed in the limbs, in the forebrain, and in the neural tube. And what people have seen is if you just inject the promoter and the gene itself, you don't get this complex expression pattern in the limbs, in the neural tube, and in the brain. However, if you start injecting much bigger chunks of DNA, you start getting these complex expression patterns because there are DNA sequences called enhancers that tell this gene to turn on in the limb, in the neural tube, and in the brain. And these sequences are called enhancers and function as the promoter of the promoter, telling the promoter when to turn on and what tissue to turn on and at what levels to express the gene itself. So why do we care about these enhancers, these regulatory elements? Well, if you have mutations in these regulatory elements that tell genes when to turn on and when to turn off, that could lead to phenotypes. And some phenotypes that could be involved with this is just basic human variation, how we're different from one another. Another is human disease, for example, limb malformations, which we'll discuss a little bit later on, but there's a lot of other diseases that could be caused by mutations in enhancers. And finally, morphological differences between species. It's thought that a lot of differences in morphology between species could be due to changes in gene regulatory elements. So, again, why do we care about these? Well, currently now with sequencing technologies becoming extremely cost-effective and really revolutionizing how we do things in genomics and in clinical evaluations and so forth, as you can see from this graph, the amount of sequenced genomes is rapidly increasing, and the sequencing cost is rapidly being reduced. And so we can make millions of genomes, and in the future we'll have a lot of genomes being sequenced. And the problem there is that when we sequence a genome, only 2% of it is actually coding, and 98% is non-coding, and where these regulatory elements may lie. So just to illustrate this through the slide, only these two base pairs will actually be coding exons, which code for a protein. And all the rest of the genome that we sequence is actually non-coding, where a lot of these enhancers and regulatory elements may lie, where we still don't really get a good understanding of their function and what they do. In addition, in terms of variation, whenever we sequence a genome, we get on average 3.5 million variants, and only a very small number of them is actually in coding exons, 0.6%. And again, to illustrate this here, this would be the small little red part of the C here in terms of having variants in exons. And so we have this vast genomic space to search for regulatory elements, and in it, most of the variation that we get from sequencing genomes lies, and we really don't have an understanding of it, of what it does. And in addition, non-coding regions, as I mentioned before, could be a major source of variation and disease. A lot of genome-wide association studies have found a lot of the associations with variants in these regulatory elements and human disease. Many Mendelian studies, classical ones, still do not find a lot of mutations in coding sequence, and so there could definitely be mutations and have been found mutations in regulatory elements that might lead to phenotypes. And as I mentioned, the human genome contains thousands, millions actually, of regulatory elements and enhancers, and so we are still not that great in trying to tie mutation to the phenotype. And one of the main reasons for that is that we don't have this regulatory code. So unlike genes, when we know the regulatory code, and for example, CCC codes for proline, and we know the consequences of nucleotide changes to that code, you change here a CTC to a CCC to a CTC, proline changes to a leucine, and that could lead to some sort of phenotype. But for regulatory elements, we don't have that. So even if I was to find a sequence that I know is an enhancer and I see a nucleotide change in it, I really don't know what it's doing on a phenotypic level at this stage. And so that's a really big problem for us in the field. And we can find variants now in non-coding space, but we really don't have a good understanding of what they're doing and how they lead to phenotypes. And just to illustrate how big of a problem this is, I thought I'd show an example of probably the most common example of an enhancer that actually leads to a human phenotype, in this case limb malformations, and just how much little we know about how mutations in this enhancer actually lead to human phenotypes, in this case human disease. So here we have a gene called sonic hedgehog, and mutations in the coding region of this gene right here lead to various diseases, various phenotypes, including holoprosencephaly, brain defects, cyclopia, kids are born with one eye, limb malformations, a lot of bad things in terms of the coding mutations. And then 1 million base pairs away, in an intron of another gene, so this is a non-coding sequence that is 1 million base pairs away from the gene that it regulates, there's the sonic hedgehog limb enhancer that tells sonic hedgehog to turn on in the limb at a specific time point. If we delete this non-coding sequence in mice, we get mice that have truncated limbs, as you can see here, compared to their wild-type litter mates, as you can see on the left of this. So this clearly is an example that a non-coding regulatory element, when you remove it, leads to a functional phenotype, in this case, truncated limbs. In addition, if we sequence patients who have an extra finger, everybody who has an extra finger in these families here has a point mutation in the sonic hedgehog limb enhancer, and you can see that these point mutations segregate nicely with the phenotype itself. There's now about 18 different point mutations that have been found in humans in this enhancer to actually lead to limb malformations in humans. And so we can use these point mutations to try and see, do we get a better understanding of phenotype from genotype? And as I'll show you, we're still very far away from that. So this is an example of a big Swedish family that came into our lab that had an extra finger. And so the first thing that was done was sequence this family for the sonic hedgehog limb enhancer. And when we sequenced it, because the phenotype looks similar to these other mutations that I showed you, we found a 13 base pair insertion in all the affected cases. So all of these guys had these 13 base pair insertion that was added to the enhancer. And so the next step, and this is in all the affected members up there on the top. And so the next step is to try to characterize what this 13 base pair mutation does. Now I would expect that a 13 base pair insertion would lead to a much more drastic phenotype than a point mutation, yet the phenotype that we observed in this patient is exactly identical to the point mutations that I showed you before. So again, as I said, the next step is to try to characterize functionally what this mutation might be doing. One thing we can do is look at what potential transcription factor binding sites changes are there. And you can see here below that a lot of binding sites were actually gained by having this 13 base pair insertion, but not lost. And so it could be that there's now a lot of things that bind to the sequence that shouldn't bind. But this is all in silico predictions. We really don't know what it might be doing in terms of a phenotype. And so the next step is to try to test this functionally. And the way we do this in the lab is we have this enhancer assay that we and a lot of other labs use, where we can clone, for example, the wild-type sonic hedgehog limb enhancer sequence here in front of a minimal promoter. A minimal promoter is like a light switch that's always off. If it has an enhancer right here in front of it, that will turn on the minimal promoter, and that will turn on the reporter gene right here, and that will be something we can assay, the reporter gene being something like a GFP, LAC-Z, luciferase, or so forth. We can measure or look at the expression of the reporter gene and see where it's expressed. And so we can take the wild-type sequence, as we do here for this enhancer, and in this case generate transgenic mice, and see where this enhancer is expressed, expecting it to be expressed in the limb, similar to what I've shown you before. And then we can also take the 13 base pair insertion mutation right here. So we're taking exactly the same sequence as above, but now with that 13 base pair insertion into it, and putting it inside the same construct in front of the minimal promoter and reporter gene, and asking the question, is there a difference between them? Does this give us, the one on the top, give us a different expression pattern versus the one in the bottom in mice? And so this is the results for this. So for the wild-type sequence, we see expression exactly where sonic hedgehog is supposed to be expressed, as you can see here, in the bottom of the limb right here. Now, what do we get for the for the 13 base pair insertion? When we do that, we now see that sonic hedgehog is potentially not only expressed right here, but also expressed right here in these regions over here, suggesting that that's why we get this extra finger here, because it's not only restricted to this side of the limb, but now also to the other side of the limb. And so this is great. This functional assay can give us a sense of what's happening, but it's very limited in various ways. One, each transgene has a different number of insertions of our enhancer assay vector, and so it's really not a quantitative assay. Each embryo can have different numbers of integration, and so we can't really measure expression levels for this. It can look at differences in location in terms of expression, as we see here, which is great. But that will not be the case for all enhancers. There could be differences in expression levels, quantitative differences, which we can assay. In addition, for this enhancer, there are 2 point mutations in position 402 right here, or 404 here, that have a much more severe phenotype, where not only you have an extra finger, but also the radius and the ulna are affected, and also the legs. And these are point mutations, not a 13 base pair insertion. And again, I would expect a 13 base pair insertion to have a much more severe phenotype than a point mutation, but this is not the case. And so here we actually were the ones to find the 402 mutation, and we did exactly the same thing. Take that 402 mutation, clone it right in front of the vector that I just showed you here, in front of the minimal promoter and reporter gene and test it in a mouse. And when we do that, we get something like this, which we do see ectopic expression, differences in expression than what you would see in the wild type. But it really doesn't tell me why this 402 mutation leads to a much more severe phenotype compared to going back here, where this 13 base pair insertion and the expression pattern that we see below leads to a less severe phenotype of just having one extra finger. So all of this is to say that we're really not at the point where we can really tell a phenotype from a regulatory element genotype, and that's a big problem for us in the field. So to sum this up, our current limitations is that we have tons of genomic data that we're getting from all these genome sequencing. We have a very limited understanding of the regulatory code, and even if we find mutations in known enhancers, like I've shown you for the sonic hedgehog limb enhancer, it's really hard to make sense of what they're doing on a phenotypic level. And finally, we have this very limited functional assay that really doesn't give us a good readout, a quantitative readout, and more importantly, we're testing a sequence every time one by one here. So we can only clone one sequence here while we're finding millions of variants in the genomes that we're sequencing. And so it's very limited also in terms of its throughput, that we can't use it to really test thousands of these variants and millions of these variants that we are finding in patients. And so this really needs to be improved upon. And so in order to do that, we've been collaborating in the past 5 years with the lab of Jay Shandur, and that's the work that I'll show you today in the University of Washington, and also with Greg Cooper in Hudson Alpha and Daniela Witten, also in UW. And the work I'll show you is done primarily by two very talented postdocs, one in my lab, Fumitaka Inawa, who did a lot of the experimental and functional work, and Martin Character, who did all the beautiful computational analysis that I'll show you in a minute. And so the idea was to develop an assay that can allow to test thousands of sequences for enhancer activity instead of testing them one by one, as I've shown you before. So in order to do that, what we could do is, again, start with the same vector that we have before, where we clone an enhancer in front of a minimal promoter and reporter gene. But the trick is, which Jay Shandur pioneered, was to add a 15 base pair barcode right here on the right. So this is a 15 base pair sequence that can be transcribed along with the reporter gene. So if this sequence on the left is an enhancer, it turns on the minimal promoter, turns on the reporter gene, and will make an RNA that has both the reporter gene and a 15 base pair barcode. And so that case, you can start cloning thousands of different enhancers right here on the left, and then thousands of different barcodes on the right. And with paired end reads, where you can sequence the same DNA sequence from the right and from the left, you can basically assign which barcode goes with which enhancer right here on the left. And so you can know when you build the library what barcode goes with what enhancer. And so in that case, you can clone thousands of enhancers, thousands of barcodes, know which one they are, and your readout can be RNA of if that barcode is being expressed. And so we can take this library after we clone it and stick it, for example, into cells via transfection or other ways, or stick it into mice, as I'll show you a bit later on, and then basically sequence both the DNA to see if things went into the cells or went into the mouse, and then sequence RNA to see if they're expressed, which will give us a quantitative readout whether this enhancer is functional. So the RNA gives us, is it functional? And the DNA tells us, did it go into the cells? And so if it didn't go into the cells, we can't use it for analysis. And so the readout could look something like this, where, for example, if we look at this red enhancer, we got DNA into the cell, so we know this went in. And we saw a lot of RNA versus DNA for it in these cells. The yellow one right here, we saw DNA, so we know it went into the cell, but we didn't see a lot of RNA, suggesting that it's not a strong enhancer, and so forth and so on. So using this technology, again, we can test thousands of sequences for enhancer activity and thousands of variants also. So we've published on this a few various articles, and what I thought I'll do is show you a bit of what we are doing this with this now currently in present, that is still some of it unpublished, and some of what we have published in the past, just to illustrate the strength of this technology. And so one thing that you can do with this technology is, instead of taking thousands of different enhancers here on the left, as you can see here, you can take one enhancer and just do thousands of different mutations on it. So in every position, if you have, for example, in one position in A, you can change it to a C, to a T, or a G. So you have basically thousands of different combinations of mutations that could be in that enhancer. And what that can give you is a really better understanding of where the function is, where, if I mutate, does that reduce or increase enhancer activity through this. And then we can take all this library, and again, what we've done in the example that I've shown you is gone into cell lines, as you can see here, and then test DNA and RNA and see what mutations disrupt the enhancer activity or make it a better enhancer in either case. And so we have a big project where we took 10 promoters, all listed here on the top, and 10 enhancers listed here in red on the bottom, that are known to lead to various human diseases. And the diseases are variable. You can see, for example, the hemoglobin promoters that lead to thalassemia, and TERT promoter that leads to cancer, a lot of various promoters and various diseases. And same thing below for enhancers. You can see a lot of various enhancers that are known to, mutations in them are known to lead to human disease, including the sonic hedgehog limb enhancer that I mentioned before in terms of that. So we take all these enhancers and all these promoters, make tons of mutations on them, and then clone it into this library and see how the mutations affect the enhancer activity by measuring the barcode that's associated with the mutation. And so I'll just show you one example for this, and this is for the TERT promoter, the telomerase promoter that is known to be associated with a lot of cancers. So the TERT promoter and mutations in it are actually extremely common in some types of cancer. So mutations in either position minus 146, a G2A change right here, or mutation in position minus 124, a G2A mutation there, either one of these are extremely common in some cancers. So in 83% of glioblastomas, you would find either the minus 124 or minus 146 mutation. 71% of melanomas will either have the minus 124 or minus 146 mutations. So these mutations, either one of these two, are extremely common, and what they do is actually activate the TERT promoter, make telomerase active, which allows for cancer progression. And so we decided to see how mutations look at this promoter, and took basically the promoter, cloned it, and did a lot of mutations on it to see how they affect the activity of this promoter. And this is the output that we get from these studies. So you can see right here, this line in the middle is sort of the wild type expression. And so anything that goes above this line means that something is more active, and anything that goes below this line means that it basically reduces the enhancer activity, as you can see here. So for example, these two mutations right here, this one and this one right here, are actually the strongest mutations that affect the most in terms of enhancer activity, have the highest effect. And both of these are actually activating mutations that activate the enhancer. And surprisingly enough, these are exactly the same two mutations that I mentioned here, minus 146 and minus 124, which are the activating mutations that you find in glioblastomas. And so these ones serve as a great control to show that our assays can find that mutations that have a phenotypic effect. But in addition, you see a lot of other mutations that are activating, like these ones or these ones over here. And some patients actually don't have the two that I mentioned before, minus 146 and minus 124, but may have these other mutations in these other activating positions. And so having this data set, we'll hope, will be very useful for clinicians that can go and look at this data set if they find a new mutation and see what it does on a functional level in terms of the activation of the TERT promoter. In addition, there's a lot of inactivating mutations. This region in particular here is of extreme interest, as it actually reduces the promoter activity of TERT. And so if we can figure out, for example, what transcription factor might be binding here in this region over here, and we might be able to use an antagonist for it as a way to treat patients who have these activating mutations. If we can mess up the binding of a transcription factor to this region right here below, you will get lower activation levels for TERT, and that will counter the higher, might be able to counter the higher activation levels that you get from these mutations above. And so we hope that having this map of mutations, not only for this promoter, but for a lot of other promoters that are known to cause disease and a lot of other enhancers, we will be much smarter in terms of understanding what these mutations are doing. And in addition, we hope that the community will use these data sets to predict and build better computational predictions of how mutations in gene regulatory elements could lead to phenotypes, and what specific mutations can really have an effect on a certain promoter or on a certain enhancer in this case. We've tried a little bit to do that, and I'll show you one example, and this is work by Martin Kircher, where he took, for example, the TERT promoter and tried to see, can he predict where the causative and the strong mutations are based on various tools that are available currently, like conservation tools like CAD, FunSeq, and ENCODE data sets, and so forth. And I won't bore you too much with this, but what you can clearly see from this is that we're still not there yet, that the current available annotations and scores are currently insufficient to really give us good predictions of how these mutations affect the promoter activity here and also in the other promoters and enhancers that I've shown you. And as I said, we hope that having this data set can be helpful in terms of trying to better predict what these mutations are doing in the future. So this is what I've shown you is taking one enhancer and basically doing tons of different mutations on it. Another option, if we don't know a certain language, is to start building a language from scratch and seeing what works, what doesn't. And so, for example, taking a synthetic sequence and starting to throw, for example, different transcription factor binding sites. So, for example, in this illustration here, if this blue line here is a transcription factor, we can take a neutral sequence like this one here that we know is not an enhancer and throw a blue transcription factor on it, let's say, a green one here and a red one here, and ask the question, does this make it an enhancer or not? So by sort of building a language from the ground up and asking different questions, we can try to get a better understanding of the regulatory code and the grammar that is used for this code, and through that hopefully learn more and again get a better understanding of genotype and phenotype in terms of regulatory elements in this case. And so for this project, we took 12 different liver transcription factor binding sites, and the main idea is, and you really don't have to understand everything that's in this slide, is that we took 12 different transcription factors that are known to be active in the liver. This is the list of all these 12 right here. And for each one, we took a certain sequence that we know that they like to bind to, as you can see here. We then generated 5,000 different sequences that are 168 base pairs long on 2 neutral templates that had these transcription factors, and I'll walk slowly through this. So basically, we took 2 sequences that are neutral, that we know are not liver enhancers in this case, and then we start throwing transcription factor binding sites on them to see, does that make it an enhancer or not. So for example, we take a neutral sequence we know is not a liver enhancer, and then we put 1 transcription factor binding site right here on the left, on the right, and ask, is it an enhancer or not? Or we put a different, the same transcription factor but now on the left and ask, is it an enhancer or not? Or we can put, for example, 2 different transcription factor binding sites, or 8 different transcription factor binding sites of the same transcription factor. So each time we're taking just 1 here, for example, FOXA1 right here, and just throwing it on the neutral sequence and asking the question, is it an enhancer or not? And we use 2 neutral sequences so we can compare them and see, do we get similar results. We can also take 2 different transcription factor binding sites. So for example, we can take FOXA1 here and CBPA and put 1 basically right here on the left and another one right here on the right on the neutral sequence, and ask the question, is that an enhancer or not? We can put 2 and 2, 4 and 4, and so forth, and again ask the question, are we getting enhancers or not? Or also shuffle them and play around with the order. In terms of playing around with order, a lot of our library was devoted to that. We can take, in this case, as you can see here, 3 different transcription factor binding sites, and we put them on the neutral template, and then all we do is we shuffle them like a deck of cards to see if order is important for their enhancer activity in this case. So now we have basically this library of 5,000 different sequences, all with various different transcription factor binding sites right here, and we have barcodes that we link to those synthetic sequences. And then the next step is we, in this case, tested these sites both in cell lines, HepG2 cell lines, which are liver cell lines, and also in the mouse, as I'll show you in a second. So for the mouse, there's a technology that can be used to get sequences nicely into the liver, and that's called the hydrodynamic tail vein injection. It basically involves injecting a large volume of liquid, and 10% of the mouse weight. So if a mouse, for example, weighs 30 grams, that's 3 mLs, in a very short period of time into the tail vein of the mouse, 4 to 8 seconds right here. This is equivalent, for example, to injecting 8 liters of liquid into me at 4 to 8 seconds. And what you get from that is very specific liver expression, as you can see here, where somebody injected GFP into the liver of a mouse, and you see a nude mouse in this case, and you see only expression in the liver, but very less or hardly any expression anywhere else. So this technique is great for getting DNA into the liver and testing things in a mouse liver, but it's not good for other tissues. So if you're interested in the liver, it's a great technique to use. If you're interested in other tissues, it won't be that great to get a lot of DNA into the liver. And the expression of the DNA is usually the best at 8 to 24 hours, as you can see right here. And so what we do in this case is, I'm going to go back to here, is we take this library, inject it into a mouse. 24 hours later, we get DNA and RNA from the mouse, measure that, and then check for the enhancer activity, in this case, to see which synthetic sequence works, which one doesn't, and through that hopefully learn rules about these regulatory elements. So going back here forward, the first thing that we can observe is that the more binding sites you have, so this is going from 1 all the way to 8, the more binding sites you have, the stronger you are in this case. And the darker blue here, like this or this, means that you have more different transcription factor binding sites. So you not only have transcription factor A, let's say, but you also have B and C and D. So the more different transcription factor binding sites, the stronger you are. So overall, we see is that the more transcription factor binding sites that we put on the sequence, the stronger the enhancer is, and then the more different they are, heterotypic as we call them, the stronger you are in terms of your enhancer activity. In terms of one transcription factor binding site, what we see is that for some transcription factors, for example here for HNF1 alpha, if we go from having 1 copy of the transcription factor to 8 copies, we get a much stronger enhancer. So for all of these guys right here, the more copies we have, the stronger we are, which makes sense, but this is not the case for all of them. So in the case here below, there's a lot of transcription factors that no matter how many copies we put, we still don't get a functional enhancer, suggesting that this sort of rule exists probably for some transcription factors, but not all of them. In terms of 2 transcription factor binding sites, what was nice is that we can start looking at rules of interaction between different transcription factor binding sites. So for example here, if we have this transcription factor, FOXA1, and this one right here, NR2F2, if we have a red line, as you can see here, between them, that means that they interact better with one another. And if we have both of them together on that sequence, we're going to get a much stronger enhancer than just having each one of them separately. So a lot of these transcription factor binding sites like to work with one another. If you have these red lines that you can see here, they basically like to work with one another. And then we can also see these black line right here for XBP1 and HNF1 alpha. And in this case, these 2 transcription factors we think interfere with one another. And if we have both of them on the sequence itself, we actually have reduced enhancer activity compared to having each one of them separately. And so we can start generating these maps of, similar to protein-protein interaction maps of which transcription factors like to work with one another and which ones do not. Everything we did up to here is all synthetic. It only exists sort of in the synthetic world. And so we want to see, are the rules and the grammar that we see here relevant to things that we see in the wild? And so in order to do that, we went to this data set generated in Bing Ren's group in UCSD, where they did a lot of genomics on the mouse liver, and in it, through it, and basically predicted about 52,000 liver enhancers that exist in the mouse. And so we use this data set to look, do we see the interactions that you're seeing right here that I showed you before? Do we see these interactions much more than what we would expect by chance in these real live enhancers that exist in the wild? And the answer is yes. So for 7 out of the 8 interactions that I've shown you, they exist much more than you'd expect by chance. So for example, if you look at FOXA1 and NR2F2, combinations of them, you see them much more in real live liver enhancers than what you'd expect by chance together, suggesting that the grammatical rules that we learned here, the combinations that we saw here, actually do exist in the wild to make functional liver enhancers. Now the last part of the library was made up of various transcription factors in order to ask about order. And we've done quite a lot of this. Almost half the library was actually tested for order, as you can see here by the number, about 2,600 sequences. But I'll just show you one example of this, where we had different sets of 8 transcription factor binding sites where we mixed them around 49 combinations. So for example, these various 8 different transcription factor binding sites, we basically shuffle them 49 different times like a deck of cards. And so for example, we could have this sequence right here below that you see here, and that has exactly the same 8 transcription factors as the one above. The only difference is the order of some of these. So for example, if you look at FOXA1, it's not here now, but it's more on the right. Or if you look at HNF1 alpha, it's not here on the right, but now it's on the left right here. And so again, we have exactly the same 8 transcription factors. The only difference is that we shuffled them around. And then we ask the question, is there a difference in terms of the activity of these? And the answer is yes. So for example, here, the sequence right here on the top is much stronger, 6-fold times difference in terms of enhancer activity versus the sequence right here on the bottom, suggesting that order is important for enhancer activity. And so just to sum up this part, not all transcription factors increase expression by having more copies, as I've shown you some of them. Even if we add more copies on the sequence, we don't get them to be strong enough. Heterotypic enhancers, so enhancers that have different transcription factor binding sites instead of having the same transcription factor binding sites, which we termed as homotypic, and tend to be much stronger than ones that have the same transcription factor all over again. So if you have different transcription factor binding sites, you tend to be a stronger enhancer. And then some transcription factors like to work with one another, and some may interfere with one another. And that order is important in terms of the transcription factors, and that it can affect the enhancer activity. And finally, that grammatical rules that we observe do seem to appear in wild, real-life enhancers. So the last part of this and of the talk and of this technology, another thing that we've been doing in the past years is trying to develop this technology to work with lentivirus. And the advantage of lentivirus is twofold. One, it can infect hard to transfect cells, so cells that are really hard to transfect, like neurons, for example. You can't use this technology unless you use lentivirus, and so the advantage of using lentivirus is that we can actually go into these cells. And the second is that it not only infects the cells, but it integrates into the genome. And so we can measure the enhancer activity of something that's integrated within the genome versus everything that I've shown you until now that is non-integrated, that is episomal, that doesn't actually go into the genome. And so it might not be affected by the chromatin marks or other things that could be involved when things integrate into the genome. And so with lentivirus, our first study was to ask the question, is there actually a difference between things that integrate versus things that don't? Most techniques that try to find regulatory elements, enhancers like ChIP-seq, ATAC-seq, and others, and no worries about which technique here is used or so forth, but most of them find enhancers in the genome, in the chromatin. And most of the characterization, just like the ones I've shown you before that we've done, is episomal. It's done outside the genome. And so we basically wanted to see, is there a difference between the enhancer activity for things that integrate into the genome versus things that don't integrate into the genome for this. And with lenti also, you can really ask this question nicely, as is illustrated right here. And so one thing that you can do here is basically take a library, and in this is a case which we've done here, of about 2,300 enhancers that we think are liver enhancers in this case. And we clone it along with the barcodes, exactly like I've shown you before. With lentivirus, we also put in on this side right here and on this side right here what is called anti-repressors that can protect it from integration effects. Lentivirus can randomly integrate into the genome, and there are chances that it might integrate next to another enhancer, and that might skew your results. That might turn on this construct and give you completely different results. Or it might integrate into a silenced region, a region that's silenced, and might not turn on at all. And so the use of anti-repressors here on either side might protect from where it integrates and might sort of give it sort of an independent environment on either side. In addition, we clone here 100 barcodes for each sequence. So if we have, for example, sequence A that we're testing for enhancer activity, we would have 100 copies of it with 100 different barcodes that go along with the ride. And so if one copy of it, for example, integrates into a silenced region, we still have 99 other copies that hopefully will not be silenced and can give us some kind of measurement. And this also allows us to get a good quantitative measurement. So now we have this library all cloned, and we make virus that can integrate into the genome, and then we can measure, just like before, RNA and DNA and see the expression of this. What's nice with lentivirus is that we can also mutate the integrase, the enzyme that allows it to integrate into the genome. And so we can make a library that has a mutant integrase that doesn't allow it to integrate into the genome, and ask the question, what expression pattern do we get from this library? So what this allows us is actually to compare apples to apples, where we can take the same exact library right here on the top and test it in a non-integrating version right here versus an integrating version right here, and see what differences do we see for exactly the same library in terms of the enhancer activity. And so here on the right, I'm showing you the results from 3 different replicates from each one. And the first interesting observation was that integrated constructs, so wild type right here is things that integrate, that have a wild type integrase. And what we're doing here is basically comparing each one of the replicates. And for example, if we compare wild type replicate 1 to wild type replicate 2, we see that the results are quite consistent and very similar between the different replicates. Here on the top, we're comparing the mutant libraries, the ones that don't integrate into the genome, and as you can see here, they're a bit less consistent from one another. The more sort of what we really wanted to do in this case is also compare them to one another, the wild type, the ones that integrate, versus the ones that don't integrate. And if you look here on the top, where it's highlighted in green, you can see that there are major differences, for example, here between wild type 2 and mutant 2 comparisons, suggesting that there are differences between integrated and non-integrated enhancers. They are quite similar. I mean, you do see similarity, but as you can see here, the similarity is not perfect, and there are definitely many differences between them. And so we wanted to see what drives these differences, and we've done a lot of computational analysis, and we meaning Martin in this case, who analyzed a lot of the differences that can be seen between things that are active in wild type, as you can see here, versus things that are inactive in mutant. So here are the things that make wild type active, as you can see here, versus things that are mutant. And one main thing that we see that made a big difference is the number of transcription factors that a sequence might have, transcription factor binding sites, based on ChIP-seq data sets from ENCODE and other places. And also we saw a lot of enrichment for things that have binding sites for June, FOS, or ELF, as you can see right here. And these are basically cofactors, so transcription factors that bind together to activate transcription. And so what we think is that cooperative binding, having different transcription factors bind together to activate expression, is a major factor and a major difference between things that integrate versus things that don't integrate, and may actually affect the expression levels of enhancers in this study. So just to sum up, we've basically have been developing this technology of massively parallel reporter assays that allows us to test via different methods, as I've shown you, either by mutating one enhancer to death and seeing how that affects its activity, testing a lot of different enhancers and seeing which works and which doesn't, and testing a lot of variations in them, or testing them in an integrated version using lentivirus or other, and then using this technology to get a better understanding of how regulatory elements and genotype may lead to a certain phenotype. And we're not the only ones. There's a lot of other groups who are using these massively parallel recorder assays. And our main hope is that the more people use these assays and the more people publish data on these assays, that will allow us to really get a better understanding of how mutations in gene regulatory elements can lead to human disease and other phenotypes through these methods that I've mentioned. And so with that, I just want to sum up and thank members of my lab right here on the left, our collaborators in UCSF here in the middle, and our collaborators in UW, in particular Jay Shandur's lab, as you can see here on the right, and our funding resource is here on the bottom. And thank you very much, and I'll open it up for questions.
Thank you, Dr. Ahituv, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question in the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, do you get more than one mutation in your saturation mutagenesis experiments?
Yes, so the way we've been doing, so the answer is yes. And so the way we've been doing these mutations now is through error-prone PCR. So we take the sequence, the regular wild-type sequence that we cloned, and we PCR it with a Taq polymerase that is prone to make mutations. And so we get a lot of different mutations in that sequence. And so technically, if we sequence a lot from the results, we might be able to get a sense also of not only just how one mutation affects activity, but how combinations of mutations can affect the activity of a certain promoter or enhancer. But we really have to dig deep and do a lot of sequencing and hope that we get the same sort of both mutations on a certain sequence or enough data that we can basically try to model how that will be.
Did you observe any rules for spacing in the synthesized enhancers?
So in terms of the synthesized enhancers, we played around, as I've shown, quite a lot with spacing, the distance between the transcription factor binding sites. And we've done a lot of modeling to see, do we see any rules in terms of the distance between them? Unfortunately, we didn't see anything in our models. That being said, it doesn't mean that spacing doesn't have a difference here. We were limited in the way that we clone these sequences to 168 base pairs, so we have a very short real estate in terms of playing around with distance. And so it could definitely be that if we had a longer real estate or thousands of more sequences, we might see an effect on spacing. But in this case, at least in our data, we didn't see any effect on spacing between these transcription factors.
We are almost out of time. This will be our last question. Why are you limited to short sequences in your synthetic enhancer and lentivirus construct?
Yeah, so the reason we are limited to short sequences, both in the synthetic enhancers and the lentivirus library that I showed you, is the way we make these libraries. Currently, the cheapest way to make libraries and to synthesize DNA is through oligos, and so we can synthesize oligos that are about 230 base pairs in size, and we can through Agilent we've been doing 240,000 of those. That are 230 base pairs in size, and so that's sort of the easiest, cheapest way to currently synthesize things. So especially in the synthesis example, where we really want to make sequences that don't exist in the wild, we'd have to synthesize things. And synthesizing oligos really makes it the cheapest way to go and test is thousands and thousands of sequences for their activity. And so it is a limitation. A lot of enhancers could be much bigger than that, and we are working on ways to trying to get much longer sequences. Other groups have been using a sequence capture, so similar to exome sequencing, where you capture the sequence of the exomes to sequence it. You can capture a certain sequence and clone it and test, and that allows you to get much bigger sequences for this. But especially for the synthesized enhancers, we cannot use that technology and really have to synthesize our own DNA. So it's a limiting factor, but I'm sure with synthesis, DNA synthesis becoming cheaper and better with time, we can improve on that and get longer sequences.
I would like to once again thank Dr. Nadav Ahituv for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 11, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon.